I would like to start this by thinking about the key ideas in this course. I tried to narrow it down to 5 big ideas, and we're going to keep coming back to them in different ways or at different levels. So I want to make sure we are in agreement on what these are. So number 1, organismal and cellular level processes can be understood on a molecular level. Number 2, molecular structure and behavior obey physical laws and can be described in terms of energy, equilibrium, kinetics, binding specificity. Maybe most of these terms may be familiar to you. Binding specificity may be something new that we're going to add by looking at the interactions between molecules. Okay. Number 3, covalent bonds and weak interactions determine molecular structure. Structure determines function. You're going to get a little tired of hearing me say that, but for every molecule we consider, that's how we're going to approach. How does structure enable function? Number 4, interactions between genes and environment determine traits, or maybe you've heard the word phenotype in biology, traits of an organism. Number 5, life processes are regulated to maintain homeostasis, and we will spend some time thinking about the difference between homeostasis for biological system. Often also people use steady state idea and the chemical equilibrium, and these are noticed, they're not the same thing. All right, so what's biochemistry dealing with, right? When we think about biochemistry, we're thinking about molecular organization of living things. On a macro scale, you see things, we experience things, we eat things, we pet things, right? But biochemist, when looking at these things, not only appreciates them for their macro scale sort of phenomena, but also can see details of molecules, how proteins can interact with DNA. This actually a structure of glycogen, your storage, carbohydrate storage molecule that's at the center of it has this little protein structure, right? And we'll be thinking about chemical transformations or metabolism. So the questions I want us to consider today are listed here. So what is this biochemistry or chemistry of life? What's life? What is it made up of? What are the components? And then as we go on, we'll think about how they function. So first question, what is life? What distinguishes living from non-living? I'm going to give you a definition. I hope you compare it to what your current working definition is, and I hope you think about whether it needs modification. So I'm going to distinguish living things by the following 3 characteristics. They can transform energy and matter. They have ability to grow and reproduce. And they have ability to respond and modify environment. It seems really simple, right? And what I want us to think about is this a good definition? Can everything be correctly classified as living or non-living using this? So maybe think about if you see something crystallizing out of solution, right? Does that qualify as life? And if it doesn't, what are the criteria that rule that out? And if it does, well, a crystal growing in solution is not living, right? So then maybe we need to modify the definition. So once we kind of define some properties of life, we want to think about composition or units. What's the smallest unit of life? Hopefully, you're thinking about cells, right? Cell is the smallest unit. This is a picture of a mammalian fibroblast, and you can see the outline because underneath the membrane there's cytoskeleton. There are these protein structures that give cell their shape and also provide force, generate force, right? So the cytoskeleton here is stained in green. There is also mitochondria in red and the nucleus blue. Okay, so I would say smallest unit of life is a cell, which is highly ordered molecular assembly they can do the three things we just talked about for definition of life. Okay. So what do all cells have in common? I'm hoping you're thinking chemistry, right? They have similar chemistry. They're made up of similar atoms. What are the most common atoms in living things? You should know 6 probably off the top of your head, right? So we have hydrogen, oxygen, carbon, plus phosphorus, nitrogen, and sulfur. Plus there's a bunch of other things that are found in much smaller quantities. Okay, we also have macromolecules, and then if you kind of step back from molecules, you're going to say, well, a cell has a boundary, right? That's what defines a group of molecules. They have to be separated from the environment, and that boundary is the membrane, right? So all cells will have membrane, and the membranes are composed of lipid molecules. All right, so then what molecules are cells made up of? What's the most abundant one? Hopefully you're thinking water, right? We're something like 70% water. And then there are 4 types of macromolecules that we're going to be thinking about, right? So proteins, lipids, carbohydrates, plus nucleic acids. And I do want you to think about what are these made up of, right? So proteins are made up of amino acids. Carbohydrates, if they're complex, long chains of sugars, they're made up of subunits, monosaccharides. Lipids are not really polymers, but they often have hydrocarbons, like a tail of hydrocarbons attached to something else. Often we're going to say fatty acid plus glycerol will give, or several fatty acids will give you triglyceride or triacylglycerol. Okay, and nucleic acids are made up of nucleotides. So the important thing about cell is that it has information, that it moves or flows, we're going to talk about. And you've probably heard this term central dogma of molecular biology, right? It describes how genetic information flows in the cell. And hopefully a picture like that seems familiar, right? So genetic information is stored in DNA that's converted to RNA, the information converted, right? Not the molecule itself. So information from DNA is converted to information and RNA, kind of a working copy instead of the storage copy. And then information and RNA gets transmitted into production of proteins, and it's the proteins that do a lot of stuff. Hopefully, if you don't have that yet, by the end of the class, you will have more sophisticated view of central dogma, where you're going to know location and sort of what molecules that are involved in this information flow look like. So this is on the right here is a picture from Wikipedia that says, oh, well, when you move information from DNA to RNA, the language of the molecules doesn't change, right? So that's transcription. Nucleic acid to nucleic acid gives you transcription. But then when information flows from RNA to protein, that's going to be your translation. We're switching from the language of nucleic acid to the language of amino acid, right? And then often there's subsequent modifications to the protein before it's actually fully functional. At the very least, it's got to fold into a shape, because remember, shape is important. Okay? And often this information flow also includes an arrow from DNA to DNA. When we copy DNA, that's called replication. And at this point, it's a good time to start memorizing the terms if you don't have them down, right? So that's how genetic information is transmitted from generation to generation. All right, before we move away from information flow, I want to ask you, is genetic information the only kind of information that cells have, right? And hopefully you're saying no. Well, what other information is there, and how does that flow? At the level of cell, information is molecules and also shape. So when these proteins fold and do stuff, they have certain shape. And often changes to shape is what transmits information. And again, it's going to take us a little bit to get to that idea to where it seems natural. But for now, just kind of keep that in mind, that genetic information that's encoded in the order of nucleotides or bases in the nucleic acid, that's not the only type of information. And we're going to think about other types as well. All right, so the last question is, well, what are different types of cells? If you're a biology major, I expect you to know 3 types, right? If you are not, or if you're a little rusty in your basic biology, make sure to look this up. You should know about prokaryotic cells, shown here on the left, and eukaryotic cells. And actually, this picture is a little misleading because generally, eukaryotic cells are larger. The scale here is, this one doesn't show the scale. Prokaryotic cells are smaller in general, right? And these prokaryotes are your bacteria. What you're looking at the top here is electron micrograph. So cells were kind of frozen, sliced really thin, and magnified a lot using electron microscope, right? And what you see is in the prokaryote, there is not a lot of structures visible, and there's really kind of a light and dark here. On the diagram, you can see a little better that really you have a DNA, which again is a little misleading. This should be attached to a plasma membrane. Nucleoid or DNA or bacterial chromosome doesn't float unattached. It generally stays attached, right? And there is, just like any cell will have plasma membrane. When you look at the eukaryote, you're going to see multiple membrane-bound compartments, right? And so the main difference between these 2 types of cells is their structure, the complexity and structure and organization, in that eukaryotes have membrane-bound organelles, which are internal compartments that allow cells to specialize for particular functions, some parts of the cell. Once you create compartments, what's the downside? Right now, you have to worry about moving things in and out of compartments. And you're going to see, as we discuss some metabolism, that actually is going to require extra energy, this extra transport. And so what you gain in efficiency by, say, optimizing pH of solution in the particular compartment, or by sequestering your nucleic acid away from kind of metabolic processes that generate reactive oxygen species and could damage DNA, right? This compartmentalization the eukaryotic cells have is good for sort of specialized functions, but it's going to require energy now to move things across membrane. So you can start thinking about cellular processes this way. I do expect you to know basic compartments and the differences. You should be able to label compartments and describe their basic function, right? So in eukaryotes, plasma membrane, cytosol, very important to know mitochondria. A lot of metabolism will happen in these. The job is to generate ATP, right? Nucleus stores DNA, and that's where transcription is going to happen, right? Because that's where DNA is. Golgi is going to be in ER, are involved in producing extracellular proteins and moving things out for export, whereas lysosome will generally work for degradation function, for like removing kind of basically cell waste. So these are some basic ideas. To check your understanding, I'd like to end this lecture with a quote. And what I'd like for you to do is to evaluate the statement the author is making in the light of the definition of life we've given. The quote is from an oldish paper. Microbial dominance is so pronounced that fecal pellets may be considered as living organisms. They consist largely of living cells, they consume and release nutrients and organic matter, and they serve as food for animals higher on the food chain.